Now, for the last problem that we'll be solving, showing you how it is done here in our video, it's problem number 4. It reads this way. In cucumbers, orange fruit color is dominant over cream fruit color. A cucumber plant homozygous for orange fruit is crossed with a plant homozygous for cream fruits. The F1 are intercrossed to produce the F2 generation. And letter A will give us this question. Give the genotypes and phenotypes of the parents, the F1, and the F2. Show the process using a Punnett square or the fork method or fork line method. So again, the first thing that we have to take note of or do would be to determine what are the alternative characters or traits, and also which one is the recessive and which is a dominant, and then give them the appropriate letters. Now, starting with that, we're already given in the first statement which will be the dominant and which is the recessive. So we already know that the orange is the dominant and the cream color is the recessive. Since we were not given any letters, let's provide our own. Let's just make use of the letter big D for the dominant and the small d for the recessive, just to make it easier to distinguish between the capital and the small letter. And this would lead us to three possible combinations. Again, we have the big D, which is the homozygote dominant. The other one is the heterozygote D, and also the homozygote recessive D. And we all know here that the big D is the orange, the big D small d is the orange, and also the small d would be cream colored. Okay, so with that, let us now proceed with the parents. So it is described that cucumber plants homozygous for orange fruit, and that is the first parent. It is homozygous for orange fruit, which leads us to the fact that it is a big D because it is homozygous for the dominant gene. So that's the first parent. The second parent would be a plant homozygous for cream fruits. So the only way that cream fruits can be expressed or manifested or shown is that it will be small d, it is homozygous as well. And that leads us to this genotype where the big D homozygote dominant is crossed with the small d, which is the homozygote recessive, the orange and also the cream. So we're done with the phenotypes and the genotypes of the parents. Now, looking at this one, we have to go to the F1 generation. Just by looking at the parents, the first and the second, and then combining them in our mind, we already know that the F1 generation would be a heterozygote, and then that the phenotype will be orange, and that's done with that makes make us it's F1 now is done, as we can see here, the big D, small d, and also the orange. Now, just to make sure, just to show us how big the small d or the heterozygote is produced, separating the two big D's into separate gametes. We have the big D and big D over here, and the genotype of the cream, separating them into two gametes. We have the small d and the small d, and the first pain is a heterozygote, the second pain is a heterozygote, the third pain is a heterozygote, and the fourth pain is a heterozygote, which leads us to a genotypic ratio of 4 out of 4 heterozygote big D small d, reducing it into 1 big D small d, and a phenotypic ratio of 1 orange as well. Because 1, 2, 3, 4, there are 4 orange colored progenies, and reducing this, it becomes 1 orange progeny. So this will be giving us the genotypic ratio and the phenotypic ratio, which we will be using in the letter B later on. So we're done with the P, the parents. We're done with the F1 generation. Let us now proceed with the F2 generation. To get the F2 generation, it was mentioned that the F1 is intercross, which means that you get one of the F1 and then you will cross this with another of the F1. Since the F1 is heterozygote, you need to cross this with another heterozygote from the F1 generation as well. And this will lead us to what uh, genotypes and phenotypes. Now, in order for us to answer that, let's go back to the Punnett square and separating one of the F1 using the cross, separating the gametes. We have a big D and a small d, and then the second parent of the F1, the second F1 generation use as the in the cross. You have to separate the gametes. It becomes a big D and a small d. So the first pain will be a homozygote dominant big D, and then we have heterozygote big D small d, and the third pain a heterozygote big D small d, and the last pain is a homozygote recessive small d and small d. So we have what kind of ratio? We have a genotypic ratio of 1 big D 2 big D small d and 1 small d small d, as you can see in the Punnett square over here. And the phenotypic ratio would be, what is the phenotype of the big D? Orange. The big D small d, orange, and the other one is orange. So we have 3 oranges here and then 1 cream. So the phenotypic ratio is 3 orange is to 1 cream. And we'll be using that in the letter B. So that is how we are done with getting the genotypes and the phenotypes of the parents, the F1 and the F2. Proceeding with letter B, what are the genotypic ratio and phenotypic ratio of the F1 and the F2? Since we have already done that a while ago, we'll just lay it all out. The genotypic ratio for the F1 is 1 big D small d. The phenotypic ratio is 1 orange. The F2 generation genotypic ratio is 1 is to 2 is to 1, 1 big D, 2 big D small d, and 1 small d. And the phenotypic ratio is 3 orange and 1 cream. And that answers letter B. Okay, I hope you are able to uh, get the answer or get the procedure up to this point. So let us proceed with letter C. Now, give the genotype and the phenotype of a back cross between the F1 generation and the cream parent. Show the genotypic ratio and the phenotypic ratio. Now, in this case, when we say the word back cross, actually it's already defined. This is a cross between the F1 generation and one of the parents. And the parent that is used in letter C would be cream parent. So we have here the cream parent that is crossed with the F1 generation. So this would be what the back cross would look like. It would, the big D small d, the heterozygote orange, is crossed with a small d, homozygote recessive D, which is the cream. So this is already the half of the problem because we are able now to know what would be the parent. So just take note, the back cross would be crossing the F1 generation with the identified parent. Okay, let's now get the answer. So what would be the progenies produced by the heterozygote cross with the parent, which is homozygote recessive? So the answer to this would be this. First of all, make a cross of the gene pair, and making use of the Punnett square again, you just separate the alleles of the first parent or the F1 generation, the big D and the small d, and then you separate the alleles of the first parent, which is cream, small d and small d. So you now in the first pane, big D small d. On the second pane, big D small d. On the third pane, small d. And on the fourth pane, you have the small d. Which leads us to a genotypic ratio of, if you can see here, 2 of the big D small d's, 2 of the small d here. So it is 1/2 of big D small d is to 1/2 of the small d small d. So there would be half of the progenies heterozygote, half of the progenies would be homozygote recessive, and reducing that it becomes 1 big D small d is to 1 small d small d. So it's a 1 is to 1 ratio. In terms of the phenotypic ratio, you have 1/2 orange and 1/2 cream, and then this will also reduce into 1 orange is to 1 cream ratio, a 1 is to 1 ratio. And that will be answering letter C. So I hope you got the answers over here. Let's now proceed with the last of the question. Letter D. Give the genotype and phenotypes of a back cross between the F1 generation and the orange parent. Show the genotypic ratio and the phenotypic ratio. So in this case, it's a back cross between the heterozygote, which is the F1 generation, with the orange parent, which is the homozygote dominant. Okay, so that's another type of the back cross. And that leads us to this, a big D small d orange cross with a big D orange, and that will be the back cross. Let's now proceed and get the answer. So what kind of children will you get? So again, the first step is to make a cross of the gene pair, and you can make use of the Punnett square. You will need to separate the alleles of the first of the F1 generation used in the cross, and you will get a big D and a small d. And then you will need to separate the alleles of the parent that was used in the back cross. You have a big D and a big D. Combining them together, you will now have a big D, you have a big D, you have a big D small d, and also a big D small d. So this will lead us to a genotypic ratio of half of the progenies is big D, while half of the progenies will be a heterozygote, big D small d. Reducing that, it's a 1 is to 1 ratio, 1 big D is to 1 big D small d. But the phenotypic ratio will be quite different because you have 4 out of 4 orange is to 0 out of 4 children that would be cream colored, because the big D is orange, the big D is orange, the big D small d would be orange, so this will be orange as well. So 4 out of 4 children are orange, 0 out of 4 children will be cream. Thus this will leave us to a one orange phenotypic ratio. Okay, and that ends our discussion for the whole of our discussion over here. So I hope you were able to get all of the procedures here, and I'll see you in the next video.